Hello, everybody, and welcome to the University of Melbourne 3-minute thesis grand final. Who's excited? Woo! Before we get too excited, could I please ask everybody to either mute or turn off your phone? I'll just play a bit of thinking music for a second while everybody does that. Do-do-do-do-do-do-do-do. So I would like to begin by acknowledging that we are on the unceded lands of the Wurundjeri Woiwurrung people of the Kulin Nations, and I'd like to pay my respect to their elders, past and present, and of course to all First Nations people joining us today. And I'd like to recognise the unique place held by Aboriginal and Torres Strait Islander people as the original owners and the custodians of the waterways and the land of this Australian continent. And in the spirit of why we are all here today, I would like to acknowledge our First Nations people as our very first storytellers, because that's what we're here for today, some really excellent storytelling. I'm Associate Professor Jen Martin. I founded and lead the Science Communication Teaching Program here at the university. I'm also Dr. Jen on Triple R. If you're a Triple R listener, yay, good on you. I love talking about science on the radio. And one of the many groups that I have the great privilege of working with here at the University of Melbourne is the RDU, the Researcher Development Unit. And the RDU is here to serve all members of the University of Melbourne community. And what they are there for, or what we are here for, I should say, is to provide meaningful development opportunities, because we believe that researchers who are supported and who are encouraged and who are given opportunities to learn and develop in all the ways they need to thrive, and thriving researchers are able to do work that actually makes a positive change in the world. And that's what we're here for. So the RDU runs many events, including today's 3-minute thesis grand final. If you don't know yet about all the wonderful things that the RDU does, please go to rduevents.unimelb.edu.au, but not right now. You've got other more important things to do right now. So I think we should begin by congratulating our finalists who are sitting here in the front. Woohoo! I imagine you all know making it to the grand final of a 3-minute thesis competition at any university is a huge achievement. These speakers are all extraordinary and have been through the process of having to speak at heats. They've all been selected to be part of this grand final, and they are going to be sharing their research with us today. And I've had a sneak peek of these talks, and you are in for an absolute treat. So in case you don't know, the 3-minute thesis has been around since 2008, and it started at the University of Queensland. If you haven't heard the story of where it began, very briefly, let's imagine Queensland was in pretty severe drought at the time, and everyone was asked to limit their showers to 3 minutes. And the government sent out little egg timers to everybody with a suction cup on the bottom that you were meant to suction cup onto the wall of your shower so you didn't have a shower for longer than 3 minutes. And Emeritus Professor Alan Lawson, who was the Dean of Education at the time, the Dean of the Graduate School at the time, I should say, rather than singing in the shower, hatched this idea while having a shower. Well, if I can get myself clean in 3 minutes, then surely a PhD student can explain their research in 3 minutes. Not sure quite the analogy there, a little bit different. And, 180 seconds, it's not very long. Not very long to explain complex research. But, being concise, being able to explain research in accessible, inclusive language, I think is one of the most important skills that researchers can have. So I am a huge fan of this competition. And clearly, many other people are also fans of the competition because the 3MT is now held in more than 900 universities in more than 85 countries around the world, which is pretty extraordinary. So, on to our event. Today's grand final, we have some very important jobs. We need to choose the People's Choice Award, and that is going to be your job. I will tell you later exactly how you are going to vote, or not. I'm not going to tell you who to vote for, but I'm going to tell you the process for voting. And of course, we need a runner-up and we need a winner. And the winner of today's competition will go to the 2023 Asia Pacific 3MT competition semi-final. So it's a big deal. The person we choose today will be representing the university on a very big stage. Speakers know that they are limited to 3 minutes and 1 slide. They have a lovely big screen down here, which is going to help them keep track of the time. And they know that if they speak beyond 3 minutes, they get disqualified. We don't want that to happen. Now, of course, to be able to award our prizes, we need judges. And we have 4 incredible judges with us today. They've all got many accolades to their names, and I'm not going to read out a long list of their achievements. You'll be relieved to hear. Suffice it to say, they really are extraordinary, and I think that's lucky because they've got quite a job on their hands today, having heard these talks. So let me introduce you. First, we have Professor Kat McFerrin. You're going to wave to us, Kat. Thank you. Oh, yeah, let's give Kat a clap. Kat is an international expert on the topic of music, music therapy and adolescence. She's the head of the Creative Arts Therapies Research Unit and the director of the RDU, the Research and Development Unit. Next to Kat, we have Dr. Sarah Young-Spencer. Thank you, Sarah. Sarah is the Education Innovation Fellow for the Faculty of Business and Economics. Her research focus includes the scholarship of teaching and learning, curriculum internationalisation, strategic management accounting, organisational learning and corporate governments. And I didn't stuff up any of that. Oh, I did stuff it up. Oh, no. I thought that's what I said. Anyway, moving right along. I am not being judged today, so it's all good. Then we have Dr. Ashley Dungan, who's going to wave at us. Thank you, Ashley. Ashley is a research fellow in the School of Biosciences, and her research interests are in the function of microbes in complex communities and how those microbes can be manipulated to benefit the system that they are in. Now, everyone wants to rush over and ask Ashley more about her research. So she's worked primarily on coral reefs, but also in soil, native Australian mammals, crickets, turtles, you name it. Ashley's been there. Outside of science, Ashley volunteers for Seeing Eye Dogs Vision Australia, and you can see she has 2 very important participants today, Ocean and Kelly, who look like they've already fallen asleep, which is probably good, right? That's what they're meant to do. Ashley's also an avid weightlifter, so if you want to talk about microbes or weightlifting, talk to Ashley. Last but not least, we have a very wonderful 4th judge, and I'm saying that because he's one of my closest colleagues. We have Dr. Michael Wheeler. Michael is a previous 3MT winner who now teaches science communication here at Uni Melbourne with me. We co-host a podcast together. We have a lot of fun. It's called Let's Talk SciComm. And Michael's also a research fellow in the Institute for Physical Activity and Nutrition at Deakin University. So thank you to our judges. It's not an easy task. They have a whole lot of criteria they have to pay attention to. So part of my job today is going to be to try and keep you all interested for a little bit of time after each speaker to give them enough time to judge, because they've got quite a big job ahead of them. But it is now time to welcome our first speaker to the stage. Mickey, come and join me. And as you can see, Mickey is our first cab off the rank, which is both easy and hard. Welcome to the stage. Okay. And Mickey's talking about fungus fighting frogs. There's your clock. Okay. Over to you.
What if one day, while going for a walk in your local wetlands, you noticed it was eerily quiet? The normal sound of croaking frogs was replaced with a deafening silence. You wondered, where did the frogs go? Well, this is what happened in the 1980s, when suddenly, once abundant populations of frogs had all but disappeared. And in the last 4 decades, over 100 species worldwide have gone extinct. To put that into perspective, that is like losing all of Australia's marsupials. And the cause of these extinctions? A deadly fungus. It's called the chytrid fungus, and we currently have no way to stop it in wild populations. See, this fungus does not discriminate in its attack, which means that countless other species are under threat. And this frog, the iconic southern corroboree frog, could be the next victim. Historically, they were present throughout the Australian Alps, but now they are extinct in the wild because with no way to prevent their declines, they were brought into captivity. But keeping them in captivity is not a solution. It is only buying us time. So, for my PhD, I am doing something that has never been done for conservation before, and selectively breeding these frogs to increase their resistance to the fungus. For thousands of years, humans have been selectively breeding just about everything we can. We have cows which produce more meat or milk, dogs of all shapes and sizes, and over 7,000 different varieties of apples. So why not frogs? Selective breeding works by increasing a trait in a population. So in my case, that trait is the ability of this frog to survive infection. So to increase this trait, I am breeding corroboree frogs. But I am being selective on who gets to breed. Only frogs that possess traits that are associated with resistance, such as low infection loads, get to breed. And to do this, I'll take male and female frogs, place them together in an enclosure, and then patiently and politely wait. Then using a combination of genetics and observable traits, I'll assess their offspring for fungal resistance, and then continuously select and breed each generation to increase their resistance. So my hope is that by using selective breeding, I can save this species, but also pave a way forward for the countless other frogs globally that are facing extinction due to this deadly disease. See, while we cannot remove chytrid from the environment, if I am successful, there will be southern corroboree frogs that can survive in its presence. Because I think we can agree we have enough apples. Now it's time to prevent this iconic frog from becoming extinct.
Mickey, you're done. You can take some deep breaths. Okay, audience question. Who knew that there were 7,000 different kinds of apple? Really? Can you give us an exact number? 7,224. Is that an exact number? There must be, like, just over. Thank you, Mickey. That was amazing. I have to tell you all, I have a very, very soft spot for frogs because I always tell the story that I wouldn't exist if it weren't for a particular species of frog that lives here in Melbourne called the pobblebonk. Has everyone heard of a pobblebonk or a banjo frog? So my dad moved to Australia to do a PhD on pobblebonks and met my mum here. So I always say I owe my life to pobblebonk frogs. So I'm very much in favour of Mickey looking after the frogs. Have I given the judges enough time? Phew, you don't want to keep having me talk about frogs. I'll get very excited. All right, it is now my great pleasure to welcome our next speaker, who is Lottie, talking about identifying children with language disorder. Thank you very much, Lottie. Take your time.
Do you have a word that you know the meaning of, but you can never quite remember it when you want to use it? For me, it's crepuscular, when an animal is active at dawn and dusk like kangaroos. Whenever I want to use that word, I can see in my head that it starts with cur, but I can't get any further. We all occasionally have this experience where a word is right there on the tip of our tongue. But luckily, words like crepuscular rarely come up, so we manage to get by. But imagine if this happened to you often and with common words. It would disrupt your conversation with friends and family, in the classroom and in the office. Language is crucial for communicating our needs and connecting with others. Just look at us all now using language to share our passions with you. Language disorder is when an individual has difficulty learning and using language. People with language disorder have these tip-of-the-tongue experiences often and can have trouble understanding others. It's difficult to identify young children with language disorder. Currently, a rule of thumb used to guess whether a 2-year-old will have ongoing language problems is only correct about 50% of the time. My PhD focuses on accurately identifying young children with language disorder earlier so we can give them early support. I used data from hundreds of children who took part in research studies from when they were babies until they were teenagers. Their parents answered thousands of questions about their child. And I used machine learning methods to identify which questions best predict which children will have a low score in a language test in late childhood. I identified 6 questions we can ask parents in 1 minute that had at least 70% accuracy for identifying which children would have a low score in a language test at 11 years of age. Because parents can answer these 6 questions in 1 minute, this means it's a very low resource and scalable method for identifying young children likely to have language disorder. And 70% accuracy is a big increase from the 50% achieved by previous quick methods. There are only limited resources in research and clinical services. This improved accuracy means we can better spend those resources on the children with language disorder who really need support. Identifying young children with language disorder earlier means we can give them the support they need to communicate more effectively and to live their best lives. Thank you.
Thank you, Lottie. Well done. So when Lottie and I had a chat last week talking about her research, I had to laugh because of her choice of word being crepuscular, because as a mammal biologist, that's a very common word for me. We use crepuscular all the time. So yeah, I thought it was interesting that that's your word. Has anyone else got a word to offer us that's your word that you kind of know is there but you just can't get off? Did everyone hear that? Kay just said their word is on the tip of their tongue. Boom, boom. But yes, crepuscular is a very common word for me. But I thought that was pretty amazing to get it down to 6 questions in 1 minute. It's pretty extraordinary. We're good. All right. Thank you, wonderful judges, for not making me talk for too long. It's now my pleasure to welcome our next speaker to the stage, who is Simone, to talk about music to one's ears. No? Oh, is my list incorrect? Oh, no, sorry. I misread it. Valeria. I'm so sorry, Valeria. That is completely my fault. Simone just nearly had a heart attack and is now breathing very fast. My apologies, Simone. It would pay me to actually look at my notes properly, wouldn't it? Valeria, welcome to the stage to talk about an impossible choice.
Thank you, Jen. Blood cancer. It claims the lives of over 5,000 Australians every year. This disease has a devastating impact on all aspects of patients' lives. CAR-T, which stands for chimeric antigen receptor T-cell therapy, has revolutionized the treatment landscape by modifying patients' own T-cells to help them find and kill cancer cells. But the downside is a serious side effect called neurotoxicity, which affects the brain. It is not well understood, is unpredictable, and can become life-threatening. Patients can experience many cognitive symptoms, such as confusion or changes to language and memory. Here is an example of one of our patients who lost his ability to write and another who was unable to draw, which was confronting for them and their families. As a clinician, I spend my time talking to patients about this impossible choice that they face, whether to go ahead with CAR-T, hope for a cure, but risk this dangerous side effect, or go into palliative care. Incredibly difficult decision. This is why my research aims to deeply understand neurotoxicity. What does it look like? How does it evolve? What are the risk factors and how do people recover? My study is longitudinal, so I see patients and examine their cognition before they come in for CAR-T, and then I monitor them after treatment for up to a year. I have seen over 80 patients, and so far, reassuringly, I have found that for most, neurotoxicity resolves within a month after CAR-T. For those who still have some lingering cognitive symptoms at that stage, the daily impact is mild and completely resolves within 5 months. These findings will help patients and their doctors make informed choices about treatment. We'll be able to tell them that even when things look this bad, it is not forever. My research will help reassure healthcare regulators and policymakers to make CAR-T available to more people, to give them a second chance at a fulfilling life. I am very pleased to say that both of our patients are alive and well today, free of cancer and of any cognitive symptoms. This is an incredible outcome that makes my work so rewarding. Thank you very much.
Thank you, Valeria. Well done. I find every time I hear a 3-minute thesis talk, I'm just desperate to ask questions. And I know that's not part of the 3-minute thesis kind of, setup. But I have to ask, so you're saying that somebody can go from incredible challenges like that with something that they would, previously have taken for granted, like being able to write, and that can improve and go away in the space of a few months? Sorry. I'm probably not allowed to do this. So the answer was in the space of weeks or? Wow. Within a month. That's incredible. I just can't imagine how disconcerting it would be to have something that we all take for granted as much as being able to write and losing that. Thank you. Fascinating. Fascinating. All right. We will move on to our next speaker, who is Samriti, who is going to talk with us about some sneaky invaders. Are you okay? Have you had enough time to sort the microphone and you're okay? Yep. Yeah. There we go. Sounds good. Thank you, Samriti. Over to you.
The Greek poet Homer, in his poem Iliad, tells how the love story between Helen of Sparta and Paris of Troy led to the great Trojan War. But the most famous element of this war was rather that giant wooden Trojan horse that helped Spartan soldiers to invade the walls of Troy. What if I tell you the barrier between our gut and blood is exactly like those walls? Usually, bacteria are not able to get through this barrier, but one of them manages to do so very efficiently. And this is invasive Salmonella. It uses our own immune cells as Trojan horses to cross this gut-blood barrier. Once it reaches the blood, it secretes toxic proteins inside our cells. This leads to prolonged fever and, in worst cases, death. Even though Australia records thousands of invasive Salmonella cases every year, very little is known about how this bacteria manipulates our own immune system to its advantage. On top of that, some Salmonella groups even have a higher chance of going into the blood. My project specifically focuses on 2 such groups. Using computational methods, I am studying the DNA of over 500 Salmonella samples collected from human patients in Victoria for over 20 years. From my studies, I find that these invasive groups have indeed evolved with special features. They have a much diverse collection of infectious genes in comparison with other non-invasive Salmonella types. One of the invasive groups that I study even shows resistance against 6 commonly used drug classes that throws an additional challenge during the long-term treatment against this bacteria. To further understand the features of invasive infections, I will be studying in the lab how these Salmonella cohorts infect and survive within human immune cells, Doing this will help to identify new drug targets that can improve public health within the state and the country. And finally, we can get one step closer towards gaining victory against these sneaky Salmonella soldiers hiding in the Trojan cells. Thank you.
Thank you so much, Samriti. One of the things I love about the 3-minute thesis competition is this incredible challenge to come up with one slide to represent your research, a slide that is, accessible and appealing to look at, not too complicated. And when I first saw this slide, I just thought, what a great image. The pirate, eye patch over the salmonella. It's just really, really cool. Well done for your fantastic graphics. So last week, after I had a chat with many of our finalists, I was telling my kids about some of the amazing research that was going on, and I was explaining about this sneaky salmonella, and I discovered my kids had never heard of a Trojan horse. So thank you for encouraging me to discover that I've clearly been a very poor parent, and my children have never heard anything about a Trojan horse, and now they have. All right, we are ready to move on to grand finalist number 6, who is Cade Huckstep, talking about the vicious cycle of PTSD and alcohol use. Thank you, Cade.
I just want to, is the microphone working? Yes? Ooh, yeah, that made a sound. Okay, cool, great. Awesome, sweet. I'm not nervous at all, guys, it's fine. I want you to imagine something. It's a warm summer's night and you hear the sound of fireworks in the distance. But instead of excitement, your body is gripped with panic. Your brain seems to have made a mistake and it's reacting as though you've just been shot at. Because a few years ago, you were, and now anything could be a threat. You're experiencing symptoms of post-traumatic stress disorder, PTSD, a debilitating condition which some people develop in response to trauma. You've seen doctors and therapists, but treatment options are so limited and nothing has been able to quiet that constant panic except a cheeky beer. We all know alcohol seems to do wonders for anxiety, at least at first, but then the more you drink, the less that starts to help, and so the more you drink. All of a sudden, people find themselves stuck in this vicious cycle where their PTSD and their drinking are not only making each other worse, but also harder to treat. But an exciting new treatment is bringing hope. So this is where patients will spend 1 or 2 of their sessions with a therapist under the influence of MDMA, or yes, ecstasy, perhaps better known for its use in party settings. But this exciting new concept of MDMA-assisted therapy has shown real clinical promise for both PTSD and alcohol use. The problem is that we don't really know what's going on in the brain making this treatment effective. And looking at that in a human brain can be a little tricky. So we developed models for these conditions and these treatments using animals like rats. And this is the aim of my PhD. As you can imagine, this presents me with a couple of challenges. Number 1, what does the trauma look like? How do we get them drinking? Number 2, how does one give therapy to a rat? So what we do in our rats, we pair a harmless sound, a beep, with something scary, a little foot shock. Over time, the rats will learn to fear just the beep, not unlike someone being scared of some harmless fireworks. And so we then give them alcohol again after this and find they willingly drink it, and so this forms our condition. The therapy takes the form of essentially an exposure therapy, right? So the rats will repeatedly hear the sound of this beep, but without the shock, and eventually learn that the beep is quite safe. We found that administering MDMA to these animals just before their therapy session actually changes how quickly the animals learn that the beep is safe, as well as how much they remember this the next day. Most excitingly though, that when we give them alcohol again after this treatment, the animals who had done therapy on MDMA drank significantly less than the others, both initially and weeks later. So using this model, we can begin to really figure out exactly how this treatment is changing the brain and use this information to inform the care of the millions of humans who are suffering from these conditions, because nobody deserves to be stuck suffering.
Kate's like, whew, deep breaths. It strikes me today, one of the best things about the 3-minute thesis is just learning about all of these extraordinary research projects that are going on. And today we can all be left with the image of Mickey trying to encourage her frogs to, get up to what they need to do. I don't know if she uses some, mood music or lighting. And Kate working out how you replicate these really complex, difficult things like alcohol use and experiences of PTSD in rats. And it's pretty extraordinary to hear how one goes about studying these things. So thank you for sharing that with us. All right. We are ready to move on. Thank you, judges. They're being very efficient today. I'm very impressed. It's my pleasure now to invite Justine onto the stage with us to talk about the enigmatic sounding relationships in odd times. Thank you, Justine.
Imagine this. You've been given the task of communicating time to people all around the world in and through music. You might say, well, that's pretty easy. Just use beats per minute, like measuring a heart rate. But here's the hitch. You're dealing with music that speeds up and slows down quite a lot. It's not at a fixed speed, so you can't merely use a clock. So, what do? My research looks at what happens when we use relationships to measure time in music. Specifically, mathematical proportional relationships in odd-factored time signatures, which are numbers at the beginning of sections of music. In my research, I've got a variety of different types of music, from solos to orchestras, and how musicians treat time in each scenario is very interesting. For the soloist, a musician will be going at a particular speed, let's say 60 beats per minute. And then in the very next section, they need to abruptly speed up to 90 beats per minute. Keep in mind, 60 to 90 is at a 2 to 3 ratio. Okay, so the composer could play at 90 beats per minute, but my research has found that approach to be very dry. If instead, the composer shows that proportional relationship through numbers, the musician can feel that relationship by playing 3 beats in the time of 2. Now that gives it a far more organic approach. Of course, things get trickier the more people are involved, like in orchestras. I looked at pieces and performances where one group of musicians was playing at a totally different speed to another group. And that obviously gets quite difficult for the conductor, who needs to coordinate between the two groups. But when the proportional relationship was used in time signatures, I found that the conductor stressed a lot less, because one group was listening more intensely and they were taking their cues from the other group, and vice versa. Musicians were not only concentrating on what they were playing, but they were just as invested in what other people were playing. Performers would make eye contact, face each other, and lean in. My research shows that these particular time signatures enhance the innate intimacy found in music. It shows that what really dictates music is not time, but relationships. Thank you.
Thank you, Justine. Kate just said exactly what I was about to say. That is so interesting. How many musicians do we have in the room? Obviously, Kat. We know Kat's a musician, but are there other amateur musicians in the room? And are you all thinking the same as I am now? Oh, I want to think about this more. One of my great joys is singing in a women's choir. We're very, very amateur, but we get together, a local community choir, and having heard Justine practice, the next rehearsal I went to, I was really paying attention to partly what our conductor was doing, but also to the interactions between all of us singing. And, you know, when you hear about research projects like this, it just opens your eyes to experiencing the world really differently. So thank you, Justine. It was fascinating. Okay, we have made it to grand finalist number 8. Quite impressive on all of our behalves, I think. Excellent. So it is my pleasure to welcome Jacqueline to the stage to join us. And Jacqueline is talking about polymers in sustainable mining. Thank you, Jacqueline.
Imagine you're a treasure hunter on a sandy beach, searching for tiny hidden gems that are scattered among the sand. These gems are so small that they're hard to spot and even harder to collect. Now, to make this treasure hunt a success, what if I told you about a magical potion? A gem attractor potion. This potion contains special polymers that have unique properties that can selectively attract and bind these valuable gems, making them larger and more visible. Wouldn't that make things easier? In the world of mineral processing, fine material has always presented us with a challenge, with almost 30% of valuable fine minerals lost to the waste. My project revolutionizes this by using cost-effective polymers to unlock hidden treasures within mineral ores. But what are polymers? They're large molecules that can be tailored to have various functionalities. They can have water-loving or water-repelling properties, or even positive or negative charges. This gem attractor potion contains specialized polymers called charged polyacrylamides that act as a glue with the help of their charged nature to form clusters out of these fine, valuable minerals, such as iron, copper, and gold. This makes them more manageable and enhances their recovery by simplifying their separation from the waste minerals. I look at how big these clusters can get, how quickly they form, how we can separate them from the waste minerals, and how it can be applied to existing processes in the minerals industry. These polymers have shown their transformative powers through their targeted attraction, selectively recovering only your favorite kinds of gems. This ability makes them a crucial technique in the minerals industry. They form large clusters that are easy to recover in mineral flotation cells, where we use bubbles that can attract to larger gems compared to the finer ones. My work boosted recovery rates to nearly 100%, thus reducing the need for extensive digging from the ground. With each gem recovered, we can take a step forward to be more sustainable, and my project's impact extends far beyond the mining industry, where it embraces a balance between economical prosperity and environmental protection. As we focus on minimizing the waste generated and the ecological footprint, we can take a step forward to safeguard our precious mineral resources for generations to come. This magical potion helps make the mining process a grand treasure hunt, as we stand at the beginning of a greener, more sustainable future for the minerals industry.
Thank you so much, Jacqueline. I think everyone can agree that for our later speakers, there's a lot of sitting around getting nervous. And so to get up and deliver like that is just so impressive. And, you know, how could anyone not be excited about something that not only is going to be financially viable to make mining that is going to be both financially viable, but also, environmentally better for the planet. So I think it's very exciting research. Thank you. Still have very sleepy doggies over there. Do you think they're learning, Ashley? Do you think they're taking it all in? Everyone's thinking, imagine having a dog's life. Imagine just being able to lie back, relax, enjoy. Oh, I'm sorry I woke them up. My fault. All right, it is time to welcome Kane to the stage to talk about transforming viewers into donors. Thank you, Kane.
This is the true story of a little girl named Imani. At just 5 weeks old, she was diagnosed with a cancerous brain tumor and had to undergo emergency surgery. Her parents started preparing for the worst, going as far as purchasing a burial plot for their newborn baby girl. Fortunately, thanks to the joint fundraising efforts of live streamers through charity streams, children like Imani are provided access to life-saving treatment without incurring any financial burden. She now lives cancer-free and has been able to celebrate her first birthday, her first Ramadan, and her first New Year's. This is but one of many examples in which charity streams have helped those who truly needed it, and it's the phenomenon that I will study in my PhD. Charity streams are as simple as setting a donation goal, turning on a camera, and chatting to viewers while playing video games. This simplicity emphasizes that they are accessible to anyone, anywhere, which gives it a significant advantage over traditional fundraising methods. In my PhD, my research strategy involves examining how various engagement tools impact donations. As charity streams provide a real-time research context, I am provided opportunities to study hundreds of thousands of moment-to-moment interactions across hundreds of campaigns. The granularity afforded by this real-time feedback provides a wealth of valuable insights that were previously inaccessible or limited in scope, thus giving it a significant advantage over traditional fundraising settings. Because each and every single one of us in this room can access charity streams and participate in them, this suggests that the outcomes of this research is far-reaching and applies to a wide range of beneficiaries. For streamers, this research establishes guidelines that allows them to unlock the full potential of charity streams. For live streaming platforms, most importantly, for charities, this research provides a modern solution to the age-old problem on how donations can be solicited, which ultimately benefits the hungry, the needy, and children like Imani. Thank you.
Kane, well done. It is really hard to keep going when you suddenly lose your train of thought. That was an absolute masterclass in, nope, I know my stuff, I can do this. That was so impressive. So last week when I was talking with Kane, I'm not a gamer at all. I know zero about gaming. My family knows nothing about it. I knew nothing about this, and I was so deeply fascinated with our conversation, and Kane taught me all about how this can work, and it just seemed like a remarkable way for, you know, to support charities. So thank you for opening my eyes to something I knew absolutely nothing about, Kane. Okay, it is time to welcome Hannah to join me up on the stage. Thank you, Hannah. And Hannah is talking with us about vaccine safety. Over to you.
I'm a detective, but not your average detective. Instead of investigating crime, I investigate vaccine safety. You see, clinical trials are great at making sure that the vaccines are safe and that they're effective before they're allowed to be given to the community. But they do have some critical limitations, including underrepresentation of certain populations or difficulty in detecting very rare or very delayed outcomes. So that's why it's my team's job to keep investigating vaccine safety after the vaccine is given to millions and millions of all kinds of people. And the main way we find out about vaccine side effects is through reports. And we investigate every single report that comes through us. And most of these investigations are pretty easy. For example, we might have a middle-aged woman who tells us that 1 hour after her vaccine, she's got a sore red arm at the injection site. And that's probably related to the vaccine. Or we might have a young boy who gets vaccinated and 10 days later falls off his skateboard and breaks his arm, and that's probably not related to the vaccine. But some of our investigations are trickier. What about a middle-aged man? He gets his vaccine, 5 days later starts to feel unwell. He's rushed to hospital, they do some tests, and it's determined that he has a blood clot. So what about this? Is this related to the vaccine, or would it have just occurred by chance anyway? We found for rare events, we can't solely rely on reports to us. And that's because not everybody does report. And that's not such a problem for common things like a sore arm. We have plenty of data for that anyway. But for rare events that are medically unclear, like a blood clot, we need all the data we can possibly get to draw accurate conclusions. So my PhD is to design a new investigation method that doesn't rely on reports. And I do this by gathering data for every single person vaccinated in Victoria. And I anonymously link this data to their healthcare experiences, including trips to the emergency department, visits to their GP, or their births. And with this large list of people's experiences, I can have a look at trends more accurately than before. And I do this by having a look at trends in blood clots before vaccination and compare this with trends in blood clots after vaccination. And I communicate all of my findings to the state and national health authorities to either communicate the safety of vaccines and calm people's nerves if we don't find anything of concern, or change policy if we do, to help to make sure that vaccines stay safe for everybody and continue to protect us against nasty diseases.
Thank you, Hannah. Amazing. Is everyone now thinking back to every time you got sent a survey on your phone after you had a COVID vaccination, asking you about your symptoms? Now we know why it was so important. Yeah, Hannah's like, yes, me. Did you see, like, was that coming to your team?
Yep, it's called Vac Safety, the text messages that went to people.
Wow.
That's one of the reporting methods that we heard about side effects.
Wow. Now I feel so virtuous that I answered all of those all of the surveys when they came to me, because now I know that Hannah wanted them and Hannah did something with them. Oh, I feel very virtuous. Wonderful. All right, Oliver, you have had the toughest job today of waiting to be our very last speaker. Thank you for being so patient. I'm thrilled that your time is here. So please join me in welcoming Oliver to talk with us about cancer cells and vessels. Thank you, Oliver.
Cancer occurs when your cells uncontrollably grow, but their eventual spread to critical organs is the key driver of mortality. So it's simple, right? Understand what mediates the spread, prevent it, perhaps reduce the lethality of cancer. As simple as this process sounds, there are many perpetrators involved in this well-organized crime. I want to introduce y'all to someone you'd be familiar with. Introducing the quintessential mafioso boss, aka Kingpin of the Enterprise, aka the tumor. Now his mentality is simple. He don't want to be a product of his environment. He wants his environment to be a product of him, employing everybody possible before taking over the entire city. He sets up his stronghold in one of the local suburbs, employs his henchmen to go into the streets to do his dirty business with an offer they can't refuse, pays off a couple of cops along the way to bat a blind eye, and bada bing, one suburb at a time, king of Vessel City. Now, believe it or not, but what I just explained to you is something that researchers have been ruminating over for decades. To clarify, think of Vessel City as your very own body. Like a city has suburbs, your body has organs that are all interconnected through a network of streets known as blood vessels. Now, during cancer, a tumor, much like a mafia boss, will establish itself in one of your organs, though its intention is to spread to distant organs. But it can't, and it won't do this alone. Instead, it employs cancer cells as henchmen. They're told that if they do well, they'll be promoted to lead the new stronghold. But they can't just go in there guns blazing, or they won't last long. Remember, this is well-organized crime that we're talking about. There are also good guys in the city too. Well, sort of. You see, your immune cells, much like the police, are there to stop crime or prevent disease in suburbs or organs. However, on occasion, tumor cells, using secret messaging, can coerce these immune cells to act against us, helping prepare these suburbs or organs for the arrival of cancer so they can set up their new strongholds uninterrupted. While technology has allowed researchers to take some bird's eye images of this well-organized crime, the images, they lack resolution and the crime remains unsolved. It still remains unclear who exactly in the city's in on the job, what secret messages are being sent out, and how these signals are priming organs for the arrival of cancer. Now, my job as a researcher is simple. We got to clean the streets. With the rise of new sequencing technology, we now have the ability to profile and understand the behavior of individual cells involved in the spread of cancer by looking into their RNA. Think of RNA like a mobile phone. I'm a private investigator, and I can now tap into the mobile phones of individual cells involved in the spread of cancer. I now know exactly who they are, where they are, and what they're planning. With this information, I can intercept them using various therapies to prevent the spread of cancer and its associated burden on society. And just remember, it's nothing personal, it's just business.
Who thinks Oliver should get an extra clap for that accent? I have to ask Oliver, has that always been one of your specialties, being able to do accents, or is this a new skill you've developed for the 3MT?
When I wrote the script, when I read it in my mind, it actually came out as a New York, kind of dirty accent. I was like, I have to do this, it'll just make it.
Full character immersion. I absolutely love it. Well, they were our 11 extraordinary speakers. Can we have a huge round of applause? So fascinating and so impressive. All of you have just done an incredible job. Thank you for all of the energy and effort and time that goes into giving a talk like that. It is not easy to do. So thank you. And thank you to the audience for being so wonderfully warm and, cheering along. I just think it's been really wonderful. So now our judges, of course, have the hard task. They're the ones who actually have to attempt to come up with who our winners are going to be. Good luck, friends. I hope Ocean and Kelly can help. So our judges are going to disappear. Your job, of course, now is to vote for the People's Choice Award. So I will leave you. Obviously, there's a URL there, or you can scan that code. If anyone's having any difficulty being able to cast their vote, please do let us know. But essentially now we're going to have a little break while we let the judges deliberate. There's no tap dancing or any other dance or anything prepared for you. So please feel free to chat. The catering is not until after the prizes are awarded. I'm sorry. If everyone was thinking they'd just duck over and grab some food or drink, you are going to need to wait for a little bit. If you need the bathroom, if you go straight out this door and just kind of follow the wall around to your right, you will find that. So please chat, wander, but don't go too far. I'm guessing our judges will probably need something around 10 minutes. And when they come back, we will be announcing the winner of the People's Choice, as well as our runner-up and our first place. So enjoy, and I will let you know when we have some decisions to announce. And well done, everybody! All right, everybody, take your seats. Wow, what a responsive audience. Everyone went so quiet so quickly. Thank you. Thank you for your patience. We are going to hear from the judges in just a moment, but I need to let you know, if you haven't yet cast your vote for the People's Choice Award, you have 1 minute from now. Counting down from 60 seconds. We're already at like 58 seconds because we need to obviously close the voting so that we know who to announce as the winner of the People's Choice. So please, very, very, very quickly, if you haven't cast your vote, you've now got 30 seconds to do it. Now, before we actually announce our winners, I'm just going to briefly invite our judges to come up here and join me just to tell us what they thought. Come up, judges. Come and let everybody see your faces. So we have Michael and Sarah and Ashley with Kelly and Ocean. And Kat, I will come and stand over here. Michael, tell us, what did you think?
Thank you, Jen. It's an absolute privilege to be judging this. I feel both highly entertained and highly informed after all of those presentations. They were excellent, and it made our job very difficult. So well done to all the contestants.
Sorry, Sarah.
Okay. This is my first time to be invited as a 3MT judge, and I'm not going to lie, but I'm actually absolutely blown away by the caliber and also the determinations that you have showcased in your presentation. So I extend my heartfelt congratulations to all of you. And today, whether you win or not is not important. This is just one step in your academic and professional life. And but if you carry forward with this kind of curiosity for inquiry and your determinations to make this world a better place, and I think that you are going to make a mark in your field. So congratulations.
Beautifully said, Sarah. Beautifully said. Thank you. Ashley.
Also my first time judging, and it wasn't that many years ago that I was doing my PhD here. So I can sympathize with the time you guys have spent repeating this, possibly in the shower. But overwhelmingly, I'm impressed with your storytelling. No matter what you do next, you're going to be telling stories about the work you do, and not only could that be contribute to success in things like grants and fellowships, things you write on paper, but it's going to inspire other people who thought, oh, maybe I don't belong in this discipline, this field. Yeah, your stories are going to move people and maybe otherwise get them involved when they wouldn't have. So great job to everyone. It was a difficult task.
Woo! Thank you so much, judges. You can see how fortunate we are to have such wise, thoughtful, amazing people judging today. I think you're allowed to take your seats if you would like to. Don't have to. We can have a big party up here. So we are at the business end of the day. Everybody is no doubt excited and nervous. What's going to happen now is that I've just been handed the golden slip of paper. It's just a normal white piece of paper, but it has on it who won the People's Choice Award. So it will be my great privilege to announce that. And then we will hand over to Kat, who has the very important job of no doubt reflecting on the competition and also speaking and letting us know who both our runner-up and our winner are. So can we have a drum roll, please? How long shall I just keep you going? Thank you, you're very accommodating. I'm so pleased to announce that the winner of the People's Choice Award for the University of Melbourne 2023 3-minute thesis competition is Michaela, talking about frogs. Woo! Come up. Massive congratulations, and I can let you know that Michaela was the winner of the 3-minute thesis competition in the Faculty of Science, so she is no stranger to this stage. And I think we have a gift for you, Mickey. Woo! Well done. Let's look after those frogs. Okay, Kat, now you have the difficult job of taking over. Good luck.
Thank you. It is a very exciting moment. I'm thrilled to be the one to announce the winners, and my heart is also going out to people who are not going to be announced as the winners in this particular competition. It was very difficult. We sat around the table, we negotiated, we counted, we narrated, we told a range of compelling stories to one another in exactly the way that you are learning to do now, because that's how academic life works. Objectivity is not so easy to achieve, and often we just rather need to argue for our subjective opinions and have a good amount of data to base our own opinions on. But they are just opinions, and it could have gone in a lot of different directions. So I do want to say congratulations. Lots of interesting research. That's not surprising, but some fantastic performances as well. Great use of humour, gorgeous use of the stage, some people being very well organised and perfectly presenting exactly what they intended, and others having a bit of a casual, engaging way of getting our attention. I'm just dragging it out now. The poor things, they're all just looking at me like, can you just say the names? Enough already. We learned about resilience in school. Yeah? Okay, so that's what I'm going to do. So I'd love to thank the judging panel. It really was beautiful to talk with you and to reach these conclusions together. So the runner-up for the 2023 3MT competition at the University of Melbourne by a very small amount was Samriti Thakur. Thank you. We thought you did a marvellous job of engaging us. You were one of the smooth-moving style conversationalists, and your use of the Trojan horse was very clever. We all felt that the links that you made between your research question and what you did and what was really important about your findings, it was so clear, as well as being engaging. So it was that real combination of both that we thought was brilliant. Well done.
Thank you so much.
Okay. All right then. So, the person who is going to fly away off to the competition to represent the University of Melbourne for the 2023 3MT competition is Cade Huckstep. Congratulations. We really did enjoy the way that you were also very free in your presentation, that your passion for the topic was really clear, and also a very sympathetic and accessible portrayal of an extraordinarily complex topic. And we all felt that we understood it. I was even feeling a little bit of sympathy or care and attention towards your rats. So everybody was treated with a certain warmth in a very difficult terrain, which is difficult to navigate. So congratulations on your work. Oh, yes! There's a cup!
How to win things. Someone give me instructions. Where's a camera? There's a camera.
Excellent. You can hold on to that for a while. Thank you, everybody.
Thank you, Kat. Cade, you need your flowers.
Oh my God. I have a carpet.
I don't know if it quite works that way. Obviously, my job is to just say many, many thank yous and congratulations. And first of all, congratulations to all of our speakers. It's an extraordinarily difficult thing to give a 3-minute thesis talk. It's also extraordinarily difficult to judge the 3-minute thesis. And here I am throwing things on the ground, I had no idea who was going to be the winner, even though I've judged this competition for many, many years, because you all did such a stellar job. So congratulations to all of our speakers. Very, very impressive. Thank you, of course, to our audience for joining us and being such a receptive and responsive audience. I think that really helps our speakers. Thank you to our judges for doing the difficult bit of the day. It was really wonderful to hear your reflections, and thank you for your work. It takes a lot of concentration to judge the 3-minute thesis, so I'm guessing they're all exhausted. And looking forward to a cup of tea or whatever it is that's over there. Thank you, of course, to the RDU team who've worked really hard. Thank you to Lucy. Give us a wave, Lucy. And to Andrew, and to Ruben, who is not only taking photos today, but it's Ruben's birthday. So happy birthday, Ruben. What a way to celebrate your birthday, coming and working at the 3-minute thesis competition. It doesn't get much better than that. So please enjoy the rest of the afternoon. Congratulate your friends, your colleagues, even speakers that you've never met before. Make sure you congratulate them because it is such a big thing to put yourself forward for this competition. Thank you for joining us, and please enjoy our refreshments. Thank you.